My dear students, today we are going to see the different approaches for studying animal behavior. Animal behavior refers to the study of whatever the animals do. Ethology, it is the scientific term for studying animal behavior. That is, ethology refers to the scientific study of animal behavior. And animal behavior, it refers to everything that animals do. It includes behaviors like feeding, hunting, drinking, communication, migration, courtship, social behavior, and other behavioral patterns. So there are so many methods and ideas are in practice for the investigation of animal behavior. So these methodologies can be classified into two different approaches. They are laboratory studies and field studies. Laboratory studies is the investigation of animal behaviors in the laboratory. It may include special equipment and tools to observe the animal behavior. And whereas field studies, it refers to the study of animal behavior in the field, in the living condition. So that is in vivo. Whereas laboratory studies, it is the study of animal behavior in the laboratory condition, in vitro. So for the researchers who are interested in such laboratory studies, the first major goal of such researchers was the localization of brain functions to know which areas or structures of the brain were involved in controlling particular behavior. So these brain studies include three types of techniques. It is neuroanatomical, neurophysiological, and neurochemical. Neuroanatomical studies is also called lesion or ablation technique. That is, lesion means causing a wound. Ablation means removal. So either causing a wound or removal technique. This is the oldest but most crude method to study the relationship between the particular neural structures and behavior. For this purpose, certain regions of the brain are destroyed or their function is found out from the abnormal behavior it causes on the animal. So whenever a damage is caused to the brain, it will cause some alterations in the behavior. So the brain damage may be either larger or smaller. The larger ablation, that is the removal, may be carried out by freehand by means of a knife cut. Whereas small lesions are produced by passing electric current through platinum iridium electrodes to cauterize the area. Cauterize means to burn the skin or flesh or the neural structures with a heated instrument. Small lesions can also be created by neurotoxic chemicals like canic acid. The neuroanatomical studies, it also include stimulation methods, that is ablation techniques. It causes removal or suppression of a neural structure. So this stimulation methods, it involve in the stimulation of neural area and the impact of stimulation on the alteration in behavior can be studied. So neuroanatomical techniques help many workers to form brain atlases or brain maps, which are also known as stereotaxic atlases. That is, these are atlases where the brain structures are mapped, correlating different areas of brain to different behaviors. Presently, it is available for man and mammalian series such as rats, cats, and dogs. For the brain studies, the pioneer in this field is Pierre Paul Broca. He was a French doctor, and in 1861 he came across a patient with a head injury who had a defective speech. It triggered and it Broca, he got interested and he started observing different patients who came to him with brain injuries, and he found a correlation between brain lesions or injuries and speech in different patients. After 4 years of continuous observation, Broca concluded that there is a definite area in the brain cortex that is responsible for articulating speech. Later, using the same technique, a German neurologist Karl Wernicke in 1880 established another area in the brain which is responsible for speech. These two areas, they are known as Broca's and Wernicke's area. So here there is a comparison between Broca's and Wernicke's area. Broca's area, it controlled the infrastructure for speaking a word. That means it has control over the neck muscles and larynx. That is the voice box. The lesion in Broca's area, it causes slurring in speech, and there is difficulty in understanding the words spoken by such a person. Though the person speaks sensibly, he will try to speak sensibly, but the physical structures involved in speech, it is affected by due to the damage in the Broca's area. But Wernicke's area, it is also called as the word retriever or dictionary, because it is responsible for formation of words, sentence, relevant to pick up the relevant word for communication and to form a sentence. So if there is a lesion in Wernicke's area is caused, then a person can talk clearly, but the speech will not be articulated or coherent. It will not be it will be meaningless. It will not. He cannot form meaningful sentences, but his speech, it will be very clear, because his physical structures, there is no problem in his physical structures, coordination of physical structures. So this is the Broca's area, and this is the Wernicke's area. So later on, many similar experiments were conducted in rhesus monkey and other animals. When different blocks of different shapes were given to monkeys, uh, it was made in such a way the triangular box was with bananas. So the monkeys, they repeatedly played with the blocks, and it found the triangular box was with banana. It was repeated several times. The monkey finally learned to correlate shape and content. But when the visual cortex was removed by surgery, the monkey was not able to identify the shapes. Later on, a similar experiment was conducted by Leschly in rats. He confirmed the central role of learning behavior in 1938. He did experiments with different parts of the T, different patterns of the T maze. A T maze is one that is shown here. It is in the shape of a T. So first designed in the shape of a T by Lashley. So it is called as T maze. He removed part of the cortex and loss in learning ability was found. The loss in learning ability, it was equivalent to the amount of cortex region that was removed. So on this basis, the law of equipotentiality was proposed. It states that the animal's learning capacity is distributed throughout the cortex. Other experiments include the removal of thigh hypothalamus in mice. It is very interesting. When the hypothalamus was removed, the mice became extremely hyperphagic or aphagic. That is, hyperphagic means it starts overeating, and aphagic means it doesn't eat at all. So the animal died due to overeating or starved to death. So from this, he found appetite centers are in the hypothalamus. So hypothalamus is the region which is responsible for our feeling of appetite. So we can see the neurophysiological structures in the next class.